My work mainly focuses on finding therapeutic targets for small cell lung cancer. Small cell lung cancer is the most aggressive form of lung cancer. It is about 15% of lung cancer cases diagnosed in the United States, which is quite a bit of number. And it's the most aggressive form, and most patients have aggressive disease at the time of diagnosis. Another thing small cell lung cancer is very good at is hiding itself from the immune system. So most of the immunotherapy targets or the drugs that we have so far, they have very modest benefit when it comes to treatment of small cell lung cancer. My job and my work here that I'm presenting at AACR is to find that therapeutic combination that can enhance the efficacy of immunotherapy for small cell lung cancer. The combinations that I'm mostly working on, so in our previous work, we had found targets, DNA damage response, so the proteins that work in cell cycle regulation and DNA damage repair are vulnerabilities for this particular cancer type. So we found that if you target these proteins in small cell lung cancer, you have a better chance of killing these cancers in vitro and in vivo, both in our cell lines and in our mouse models. And following that work, which was published in Cancer Research and Clinical Cancer Research, there were several clinical trials that were formed for the DDR repair protein inhibitors. What we also found that there are other reports from other cancer types that if you have a higher DNA damage affiliation, your immunotherapy works better if you have a DNA damage response vulnerability. So our hypothesis was that if you can combine a DNA damage repair inhibitor with immunotherapy, you have a better chance of increasing the efficacy of the immune checkpoint blockade for small cell lung cancer. That was the hypothesis with which we began this project. This particular combination with DNA damage repair and immune checkpoint blockade, there has been reports in other cancer types, but our study, which is now published in Cancer Discovery, was the first study showing that this works in small cell lung cancer, combining a PARP inhibitor or a CHEK1 inhibitor, both which are like regulators of the cell cycle or DNA damage repair. If you combine that with the PD-1, PD-L1 blockade, you completely regress tumors in mouse models. And the good thing about this is we right now have FDA approvals of PD-1, PD-L1 blockade antibodies, both in the first-line treatment. So atezolizumab has been combined now is FDA approved for combination in the first-line treatment, and nivolumab is now approved for small cell lung cancer in the third-line setting. And we also already have DNA damage repair inhibitors in the clinic. So it would be very easy to combine these two going to patients. So we think that this would work very much. Our preclinical study is very, very encouraging because we see that tumor regresses and it's a very stable regression, and there is also a change in the immune modulation. So the immune microenvironment changes when you combine a DNA damage repair inhibitor with immunotherapy. The work that I'll be presenting is also combining chemotherapy. So this is a low-dose gemcitabine. And the beauty of that is, as I said, atezolizumab, which is now in the first-line setting, FDA approved in combination with carboplatin and etoposide, which is the first-line treatment. So this combination is already FDA approved. So the next step would be to see how you can increase the response of the already approved drug. So we think that combining it with the DNA damage repair inhibitor could be the next step. So preclinically, we haven't seen too much of toxicity, but it hasn't been tested in the clinic, so we would like to see how that pans out in the clinic. But preclinically, we haven't found too much of toxicity because we are using very low dose of each drug. They're so potent together that we don't have to use a very high dose preclinically. So the toxicity profile preclinically is very encouraging. But we have to see still how it pans out in the patients. The field is really going forward. Small cell lung cancer was a widely ignored disease. I mean, there was no treatment almost for the last 3-4 decades, and work from our group and from others have really taken it forward. We now know a lot more about the disease, the profile, the biology, and the therapeutics than we knew before. And we think that with the FDA approvals in the last few months, that we are going forward in the right direction. And I think we have found a way to enhance the efficacy of immunotherapy for this particular cancer type, which really needs it.